remember that gram positives and gram negatives don't always look like this. Do not forget that. Sometimes gram positives are bacilli. Sometimes gram negatives are cocci. We generally draw them like this because generally there are a lot of gram negative rods and a lot of gram positive cocci, but they can interchange. So this is a gram negative. It has an inner cytoplasmic membrane. It has an outer membrane. It has one layer of peptidoglycan. This outer membrane has lipopolysaccharides that are toxic. Remember, those are LPS. It has longer hairs that are called fimbriae. It also, some of them have a sex pilus and a flagella. Inside is a nucleoid, a region of DNA. There are little plasmids running around, ribosomes, vacuoles. These little vacuoles are little storage compartments. These little storage compartments store different things. We're just going to broadly call them vacuoles. They're sometimes called peroxisomes or lysosomes or other things. So we'll just broadly call them all vacuoles. They're just little storage pockets of food and waste, and our cells do that as well. Am I missing anything large? Peptidoglycan layer, outer membrane, inner cytoplasmic membrane, phospholipids, ribosomes, nucleoids. Good? Okay, that's most of it. So let's, oh, I'm sorry, go ahead.
If we're looking at a diagram and it's got a wiggly thing coming off of it, how do we know if it's fimbriae or flagella or a sex pilus if there's nothing to compare it to, if there's just one?
So I won't ever trick you in that way. Sex pili look weird. They look different. I'll show you kind of some pictures of what that means. And as we go along, because we're going to talk about bacterial reproduction in a couple of days. So we'll talk more about what a pilus is and what it does. But broadly, flagella, you know, they're for motility. They're always really long, and they can be on both sides. There could be many of them, but I won't trick you. Hopefully it'll be obvious that flagella are longer, and if there's two of them, the second one is the sex pilus. The sex pilus is shorter. And it's in kind of a weird spot. Yeah? Okay. It doesn't have to be at the polar ends. And then fimbriae are short. They're little hair-like structures. Lipopolysaccharides you would never be able to see unless you're zoomed way, way, way in, because they're almost as small as like the phosphates. They're very, very, very small. Okay, good question. Okay, so let's talk about what the glycocalyx is. So most bacteria, gram-positive and gram-negative, have what's called a capsule. So a capsule is outside of this whole thing. So there, we have three options. So here, I'll get rid of this one and just show you without all of our layers. So a capsule is just, it's a layer of sugar around the outside. And I won't ever confuse you. You won't ever have to determine, is that the outer membrane? Is that the peptidoglycan? Is that the capsule? Just know that most bacteria are encapsulated. Not all of them, most of them. They have a capsule. And what happens is they're just spitting out these sugars. And they spit out sugar. It's almost like mucus. It's like a mucousy substance. And so they spit this out. So is it really a layer? It's not really a layer. They're just in this mucousy substance. So they're inside of this mucousy substance called a capsule. And the more they spit out, then it's called a slime layer. And then the more they spit out and the more they spit out, it's called a biofilm. And you can have millions, billions of bacteria within one biofilm component, and it helps them dock to the substrate wherever they want to stay. And also they can share food, and they can even share DNA, which we'll talk about later. But they form like a little community in this sugary mucus. So that's what the glycocalyx is. It's also called, I know it's annoying to have two names, but they're called exopolysaccharides. Poly is many, saccharides is sugar. They're called exopolysaccharides. They're not lipopolysaccharides, they're exopolysaccharides. I know there's a lot of terminology, it sucks, just make some note cards, I can help you. Always ask if you forget if I say glycocalyx again and you're like, what the heck is that? Just 100% just throw it out there and ask. Because you'll forget all this terminology. It's like learning another language. You have to hear it 50 times before it really becomes your own language. Yeah?
You said it's inside the S layer or it's outside?
Outside.
Okay.
Yeah. So really it's like, you know, if you can imagine your intestines and you have this mucousy layer, it's just like an extra mucousy layer outside. That's all it is. It's like sugars that they spit out to help them stick to surfaces. So it's not really like a part of their actual structure. Does that make sense? Okay, so that's what a glycocalyx is. We're gonna talk more about plasmids. I'll just run through that really quick. So it's just an extracellular, extranuclear, they have a name for that, I forget what it's called. But it's an extra piece of DNA. And I know that this seems really dumb to talk about, but this is where all of bacterial genetics, not all, 98% of bacterial genetics, where medical bacterial genetics comes into play is plasmids. So we can put plasmids, you're gonna do this in one of our own labs. Our own lab, we get to put a plasmid into a bacterium and change the color of that bacterium. So it's called genetic engineering, and we get to engineer our own bacteria. So you use these little pieces of DNA, and you can put something on that piece of DNA, such as insulin. So I'll talk about it again and again when we go through our genetic engineering part, but what we used to do is gut millions, if not billions of pigs to take their pancreas. So we used to take the pancreas of pigs and isolate the insulin out of their pancreas and put it in pill form to give it to diabetic patients. Now we can grow as much insulin as we could humanly ever want. And the way we do it is with bacteria. We put the genes for insulin on a plasmid, we put that plasmid into a bacteria. Now this bacteria makes insulin for us. So yes, we use it for genetic engineering, but a scary part of it is that bacteria can share these, and all of a sudden, all the bacteria within one patient is antibiotic resistant. So they can share antibiotic resistance genes. They can share genes to make them stick stronger, to make them spit more exopolysaccharides, and they can share all those genes to also make them more virulent. So we worry about plasmids a lot, we think about plasmids a lot, we study plasmids a lot. So I'll teach you what a plasmid is and how it looks and how we make that in the lab and how you put that into bacteria. Yeah?
So can bacteria, they only share their plasmids, or can they share their other DNA as well?
Very good question. So we'll talk about that when we get to bacteria reproduction as well. But they can actually share parts of their DNA as well, but usually it's when they're dead. So they can share a part of their DNA through a sex pilus. That's not as common. But also when this bacteria dies, other bacteria can just take up their genome and make it a part of their own genome, which is terrifying also. But bacteria can do that also. Yeah, great question. Yeah.
Are the plasmids a ring of DNA or RNA?
DNA. Yes, they're all DNA. Very good question. Anybody else? Okay, cool. Okay, so here we went over these structures. We went over the outer membrane. You can read through these and what they are. So they're a lipid, and they're with these. They're called O antigens. I don't care that you know what those are. Just know that they're lipopolysaccharides. That's all that we care that you know about. And so they are highly toxic, but we'll talk about what endotoxin means. We'll talk about all this stuff as we move along. But just so you know, they're toxic lipopolysaccharides. They're only on the outer membrane of a gram-negative bacteria. So this section that we're talking about, I know there's a lot of information on here. This section that we're talking about is bacterial structure and function. So just as long as you know the structure, as long as you know the function, keep it simple for right now. We'll learn more about all this stuff as we go along. Okay, so here's Fimbriae. Here's this weird sex pilus. I'll always make sure that you know that that's not a flagella, but usually it's shorter. And then here's a flagellum, one flagella. Sometimes they have more, but hopefully it won't be confusing. I'll make it as clear as I can. So Apelles, we'll talk more about this when we get into bacterial repro, but a lot of times the code for the Apelles is on a plasmid. So that plasmid will code the proteins to make Apelles. The plasmid will transfer from bacterium to bacterium. Now this one can make Apelles. Now the next one can make Apelles. Now the next one can make Apelles, because they share plasmids. And when they share plasmids, we worry, because a lot of times we worry that when they're sharing DNA is how they become antibiotic resistant, how antibiotic resistance spreads within one patient. So one patient antibiotic resistant, they can have a non-antibiotic resistant bacteria and an antibiotic resistant bacteria, and it can share. So that's always scary. But we'll talk more about that when we talk about repro. Flagella, you don't need to know all of the structures. In some microclasses, they teach you all the structures of flagella. I don't know why. I don't think you'll ever need to know that again. I mean, hopefully it won't come up on a TEAS test. If it comes up on a TEAS test, you'll be like, damn you, Professor Nyling, I was supposed to know this. But I don't think so, so it just seems like kind of random information that you don't need. Okay. Okay, so let's talk about endospores. This you will need to know, everybody needs to know this that deals with bacteria at all, because this is a terrifying thing that bacteria can do. So they can, what's called sporulate. They form endospores. So not very many bacteria can do this. The ones that we know of that can form endospores are the ones that are in the news a lot that you've heard of. I'll tell you about what they are. But what they do is they can form an endospore, or they can form a vegetative cell. So it's mostly gram-positive, and it's what happens is you have that peptidoglycan round and round and round and round and round, thick layer of peptidoglycan. Then you have this inner cytoplasmic membrane, and the gram-positive bacteria that are able to do this, it will get thicker and thicker and thicker, and this cytoplasmic membrane will kind of shrivel up, and everything will dehydrate, and it'll run out of nutrients. And then when it runs out of nutrients, it still has the DNA in here, it still has ribosomes, it still has plasmids, it still has vacuoles. But all of a sudden, everything that's on the inside, not organelles, we don't call them organelles, all of the material that's on the inside will dehydrate and stop working. And so what it's doing is, it's like almost forming a seed. It's almost like turned into a seed of a plant or something. So what it can do is it can survive. They found endospores on the teeth of Neanderthals from millions of years ago. Not millions, 300,000 years ago, 200,000 years ago. So they found endospores from the permafrost. This is what they're worried about, the permafrost melting because of climate change, because of endospores, because of organisms that have not been around for a really long time. Now we have to worry about them again. Yeah?
Is an endospore a piece of the cell, or is it the whole cell dehydrated?
It's called an endospore when the whole thing is dehydrated. Yeah, it's not the outer membrane.
It's not like a part of it or something.
Yes, good question. Yeah, it's called an endospore now. Once
Okay.
it's sporulated, it's called an endospore. And then when it usually it will sporulate. Do you guys have an idea why it will sporulate? Like what would make something dehydrate and lose its
Temperature maybe?
Yeah, temperature, nutrients, like if it runs out of nutrients, if it's in a toxic environment. Mostly it's when it runs out of stuff, when it runs out of supplies, it'll form an endospore. And then once it has a lot of supplies, then it can become a vegetative cell again. Oh no, I should not draw this. It doesn't turn into a different bacteria. I'm aligned with that. It goes back to its normal peptidoglycan, and then its cytoplasmic membrane will come back to life, and it'll come back to life and do all its normal stuff. So it can be either a vegetative cell or a endospore. So it's called germination, actually. It's when it germinates, it becomes a living cell. When it sporulates, it becomes an endospore that's a non-living cell. But we worry about endospores a lot. And for something that we call C. diff. Have you guys heard of C. diff? Yeah, it's, it used to be called Clostridium, but now they changed the genus. It's Clostridioides difficile is the name of C. diff. And what C. diff, we worry about it so much is because that's an intestinal bacteria, it's an intestinal infection. For those of you who work in a clinical setting, have you ever experienced a patient with C. diff? Yeah, some patients with C. diff, unfortunately. It's the most foul-smelling diarrhea you've ever smelled in your life, and it's explosive diarrhea. So we worry about explosive diarrhea because you're spitting C. diff all over the healthcare setting. And C. diff can sporulate, so what happens is everything gets cleaned up, there's a couple of bacterium still laying around, they can sporulate and become an endospore, and then all of a sudden it gets onto a fomite, that person touches the next patient, and it can become a vegetative cell again, the next patient has C. diff. So it's so easily, it can be passed so easily because of those endospores. Where if it was just a vegetative cell that couldn't form spores, you'd clean up, and after a couple days it'd be gone. And maybe it wouldn't even survive the triplonophyllite, or it wouldn't survive because it doesn't have the nutrients and it doesn't have the environment where it can survive. So endospores, we're gonna do this experiment in the lab as well. We're gonna dye these and we're gonna look at endospores and vegetative cells. See, this one is sporulating right here. And this one is, that's a vegetative cell. These are the vegetative cells. This is an endospore. Yeah.
So if a particular kind of bacteria can sporulate, do all C. diff cells have the ability to do that?
Yes, all
All of
C. diff,
them.
yes. C. diff and other ones like anthrax. Have you guys heard of anthrax? It was funny because I asked my last class if they had heard of anthrax, and a lot of people haven't. Have you
Powder,
guys
white
heard of
powder.
anthrax? Yeah, I think it's age related because back when I was younger, everybody knew about anthrax because it was in the news so much because anthrax endospores were being put into baby powder, and baby powder was being put into envelopes and being sent through the post office to people like as terrorism, as like bioterrorism. So that was huge when I was younger. So everybody had heard of anthrax, but I didn't realize it's not, because I don't think it's been a thing lately. But yeah, we worry a lot about bioterrorism with things like anthrax because anthrax is deadly. C. diff is not necessarily deadly, even though a lot of people with C. diff have huge problems, and a lot of them do end up, is a high rate of mortality with C. diff, but it's not like a bioweapon like anthrax is like certain death with anthrax. Inhaling endospores is really bad. So yeah, that's another one that can sporulate, and we'll see it in the lab, some of the ones that we have in the lab. Yeah.
Can you just touch again on like how, or like when it would become an endospore, like why it would do that?
Awesome.
Other than just running out of nutrients, because wouldn't it just die in most cases if it went down nutrients?
Yeah, that's the thing is that they're really hard to kill. They're extremely hard to kill.
Like in the example you gave,
Uh-huh.
like clean up and then like when this like sporulate, why did that one sporulate on other ones?
So some would sporulate, they will sporulate if there's heat, like you said, or if they run out of nutrients, or if they run out of a lot of bacteria need like an aqueous environment, they run out of water.
So it could just happen.
Yeah, and you'll see it, like on that slide, a lot of them are turning into endospores, like I would guess some of them might die, but if they can sporulate, and then they can survive a really long time. But that was a really good point. So endospores can even survive boiling. They can survive boiling temperatures. So how do we get rid of them? So I'll talk about that in a second. Yeah.
It's kind of a similar question, but what determines if a cell can become an endospore?
So it's the species, whatever the species, only certain species can.
Do they have just a different?
Yeah, they just have a different, yes, they just have they have like a special form of peptidoglycan that can form this really thick shell.
Okay.
Yeah, great question. Yeah, only some species can. And so how do you clean? Like, how do we clean? after a patient with C. diff? How, if it can survive boiling water and it can survive a lot of chemicals, it can survive certain things, how do we clean up endospores? You have to do something called autoclaving. So that's why we autoclave, is because of bacteria that can form endospores. Because boiling isn't hot enough. You need higher temperatures. Well, how do you get higher temperature, in a higher temperature environment than boiling? You have to put it under pressure. So we'll talk more about an autoclave and what it does in the lab. But basically, to kill it, you have to put it under high pressure to increase the temperature above boiling so that it will kill the endospores. And then you have to use toxic chemicals. So sometimes we debate of like, are we harming our patient or are we treating our patient? Like what is the strength of the chemical that we need to use to get rid of the endospores? So we'll talk all about that later on too. Awesome. So moving on to what bacteria are made out of. So bacteria are very similar to one of our cells. Our cells are very, very similar to a bacterial cell. So, oh, I forgot to put out my little clicker. So bacterial cells, So bacterial cells are very similar to our cells. We're made up of mostly protein as well. We're made up of glycogen, RNA, DNA. But the only funky thing on there are peptidoglycan. And if you see lipopolysaccharides, you know that that's a gram negative. So it's that, what that little scale is a measurement of how much of each of those things make up one bacterial cell. So yeah, I just thought that was kind of interesting to look at in a neat diagram. So we'll talk a little bit more about proteins, lipids, carbohydrates, nucleic acids. It kind of goes over that in the book, so I just did it lightly. And the reason I kind of do it lightly is because we're going to go through a section on carbohydrates actually today. We're going to go through a section on lipids. We're going to go through a section on proteins. So just go through the book on that, read through that lightly, and then we'll go through each section. And definitely nucleic acids we'll talk about a ton. So here's our overall chart. That's a cool website if you want to check that out about different types of bacteria. Okay, so let's talk about this guy. So we are like hot moving along. We are pumping from here on out. So just make sure that you guys are studying a lot at home. Get together in groups. I put that discussion board, I've never used that before on Canvas, but I put it just for you guys, so that's not something I'll look at. So you guys can use that discussion board, or you can use some of my classes before have used
Discord?
What is it called? Huh?
Discord?
Discord, yes, thank you. Have used Discord, or some of them just set up text groups, and some of them have met here on campus before class or after class. Some of them have met in a coffee shop. Some of them had Zoom meetings. So get together, find your group, and remember, did I tell you guys the story? I forget always who I tell my stories to, which class, but the girls that are sitting next to me that didn't pull out their books the whole time. Yeah, so if you are with a study group, make sure your study group is a good study group. If you're gonna commit to it and like, okay, I'll show up if we're gonna get some work done. You know, pleasantries, whatever, but let's get stuff done. So form a good group. If you guys need help with that, I'm happy to help you with that. If you wanna post on Discord. One thing I wanted to say, oh, I forgot to bring that up, about our quiz is that a lot of people did well, and some people did not. So if you are, even if you're a 7, I mean, I would say no matter what, even if you're a 10er, form a study group and get moving. If you're not a 10er, absolutely, meet with me now. Let's get going now. I wanna help you get 10s on every single quiz. How can I help you get a 10? Let me show you where the information is where I pulled the quiz from. Let me help you with how I ask questions. So come to me now, not before my final grade is due and somebody's 2 points under it's heartbreaking towards the end of the semester. That's why I have a pizza party at the end, because it's just so hard for me because if you're going to nursing school, you have to have an A or a B to go to nursing school in California. If you want to go to Texas, you're probably fine with a C. I don't know. I have no idea. But A's or B's, they mostly, if you're going to go to nursing school. So a lot of my students will come to me and, oh, I have a C, but it's 2 days before the end of the semester, like, I can't help you, I can't help you study, I can't, like, what am I gonna do now? How do I help you get a B? I can help you get a B now, so just let me help you early on, make sure you work with me, and I really, really want you to go to nursing school, or medical school, or PT school, or PA school, or whatever it is that you wanna go to, so just work with me, and I will 100% help you as much as I possibly can. But let's do that now. Okay, so that being said is we have a lot of information coming up. So, just how we did right there, that took you guys 10 minutes. And I don't know if you learned, but it was just, you did it again. We talked about it at 12:30, you talked about it again. Every single time that you learn and learn and learn, then it actually doesn't become that much hard work. You just repeat, and like, oh, it's actually not that hard, I've got it figured out. So, yeah, get going. Okay, so this one, I don't want to tell you it's a hard lecture. It's not a hard lecture. It's absolutely not. But just stay with me, and we'll repeat. The first time you might not get it, the second time you might not get it, but the third time you will absolutely get it. So just stay with me. Okay, so we're going to talk about metabolism. And I'm just going to tell you a little story about this guy, because I love this guy. So his name is Hans Adolf Krebs. And he lived in Germany around the 1930s. And in 1933, first he was already a successful scientist. So he was an ear, nose, and throat physician. He loved biochemistry. He discovered already something called the urea cycle of how our bodies create urine and why they create urine. I know it seems really boring, but actually now that I've taught, when I was in biochemistry, I thought it was really boring, but now that I'm teaching it, it sounds really interesting. So I teach it in my other class about, why our bodies make urine. It's because we have a piece of protein that we don't know how our bodies can't do anything with it. It's a waste product. So very cool. We won't go off on a tangent, but he's the one who made the urea, who discovered the urea cycle. And then, in 1933, they had an ordinance where they removed all Jewish citizens from professional occupation. So he lost his job. He was no longer able to work as a physician. Super terrifying, very scary. He had a friend that worked in England at the time and asked him to come over and work with him in England. So he started working at Cambridge. All of his friends and family died in the Holocaust, horrifying. And then later on, he won the Nobel Prize, and he is the one who discovered what is called the citric acid cycle, or the Krebs cycle. And it's Hans Krebs, very cool story. So yeah, I just think it's interesting, when you hear about like, oh, Krebs cycle, it sounds awful. Like it's just, one of those things of biochemistry or regular chemistry or bio class where you maybe have heard of it one other time before. But there's an amazing story behind these scientists that have worked so hard to figure out all of this amazing science that we know about. Okay. So we're going to talk about metabolism, but let's go through some other characteristics about bacteria really quick. So bacteria, we like to think about bacteria, so what we know about bacteria so far, we know about their shapes, we know about peptidoglycan, gives us a gram-positive or a gram-negative. We know a little bit about their cytoplasmic membrane, a little bit about some of the structures that are inside. We know a little bit about how they move, not a ton. So now we're going to talk about like why they live and where they live. And so why do we care? Why do we care why they live and how they live and where they live? It's because we care when our patient is sick and how our patient got sick and how we can prevent them from getting sick and how we treat their illness. So we're going to go through bacterial ecology, like their life cycle and their status, because we need to know how they live. So just to be silly, we need food, water, love, protection. That's, my husband is always like, you need clothes, food, water, love, and shelter. So that's what bacteria need as well. They need food, they need water, they need love, they have to reproduce, and they need protection. So how do they do that? Really, when you're thinking of one bacteria, they're just living the dream. it. They just are trying to survive. They have their own little communities and their own little environment. We don't think they have any sort of nervous system and anything like that, but you know, really it's an organism that's trying to survive like every other organism on the planet. So how do they survive? They need to eat food, they need to have water around because their cytoplasm, the inner aqueous environment, helps things move around. They have to have, and then also when they're so tiny, they need to be able to move different places, so they generally move in an aqueous environment. They have to be able to reproduce, so we can talk about how they reproduce, and then we'll talk about how they protect themselves in their environment. Okay, so we talked a little bit about how they live. So when we talk about colonies, you know, that they do this really, this you remember streptococci or staphylococci or diplococci, streptobacilli, diplobacilli, you know, they don't want to be separated from their little partners. And they don't want to be separated from their little partners because it's a lonely world out there. If there's one with a good environment, they want to be surrounded by a good environment together. So they form these little colonies where they live together. Biofilms. So biofilms are that third layer where they create a giant community. And the weird thing is that scientists are studying now a lot about biofilms because almost they kind of turn into one big organism. It's really weird. Like they can share stuff, they communicate, like they have these, I forget what they're called, neuro something, I forget, where they send out like sodium molecules to communicate with the other organisms around them. Very super weird. We're not going to learn anything about that. That's when you get deep into bacteriology, but I think it's cool to learn about. Okay, so where do they live? They live on toilet seats, countertops, pet food dishes, checkout screens, doorknobs, skin, soil, seawater. They live everywhere. So I just think that's kind of cool for you to look at how many bacteria per square inch there are in every environment. They live in the deepest parts of the ocean. They live in thermal hot springs, past boiling. They live on Mount Everest, everywhere. Yeah?
When you say biofilm, you're referring back to the sugar capsules, correct?
Yes. Yep, back to that sugar environment. It's like a biofilm that they've all spit out these exopolysaccharides, or it's a glycocalyx that they spit out now that they can live in these big colonies. Yeah. Okay. So we talked a tiny bit about this, but let's go into it a little bit deeper. So bacteria can live in 3 different ways. So, well, we just kind of categorize this as all, we talk about like predator and prey relationships, blah, blah, blah. But it's important to think about too with bacteria because some bacteria are beneficial to us. And some, you know, I actually wrote that down this morning. What was I watching this morning? I was watching a video about bacteria. Why? I don't know, something in the news. I don't know. I just found something interesting this morning. But there are there are millions. I think there's something like 30 million different types of bacteria, different species of bacteria, but only 70 different species are pathogenic. So not very many species are pathogenic compared to how many there are in the world. Millions of different bacterial species in the world. But we focus on the pathogens just because this is more of a clinical based class, but don't forget they're everywhere. So the ones that are not harmful, let's talk about the commensals. So I'm just gonna give you this little annoying thing because since I was in college, we thought bacteria did nothing. We knew bacteria lived on our skin because we could grow them from the days of Robert Koch, right? We knew that there were bacteria everywhere, but we didn't know what they did. And so for a long time, we thought, oh, we have all these bacterial commensals. So even my immunology professor at Harvard was 5 years ago that I graduated, and she still calls them commensals. So the community, like virologists and bacteriologists and the microbiology, immunology community calls bacteria that live on and around us commensals. But that's not really accurate anymore. We know now that they were actually mutualists. They get benefit from living on us, but we didn't know that we get benefit from them till fairly recently within my college career. So now we know that we get all kinds of benefits from the bacteria that live on and around us, which we did not know. So we're learning more, and we'll talk about probiotics a lot in this class actually, because we'll talk about what happens when you take antibiotics. So you take antibiotics and it's wiping out your bacterial population, and what happens to you? You need that bacterial population. You know, even my daughter, she got stains on her teeth, and it affects your teeth because you have beneficial bacteria in your mouth. You need those bacteria in your mouth. You need the bacteria in your eyes. You need the bacteria in your vagina. You need the bacteria in your colon. So those are they're mutualists. So we benefit from them and they benefit from us. But you'll hear commensals, if you read about bacteria, read about commensals, really we're all benefiting, actually. But the actual definition for a commensal is one does nothing. So we get no benefit where they're benefiting by living on and around us. Yeah.
So if there's a bacteria that doesn't live on us or around us, like deep in the ocean or whatever, would that be a commensal? Or because the symbiotic, does that specifically refer to humans and their relationship to bacteria, or just a host and?
Yeah, this is
Do you know
talking
what
about,
I'm saying?
yeah, because the host, yes, yeah, so the host, so this is talking about like a relationship.
Okay, so like if it's a bacteria that's benefiting a cow, then it's still called symbiotic, even if it has nothing to do with us?
Yeah, if you'd be talking about it in relation to the cow. So if you were studying the cow, you'd be like, well, they have a symbiotic or a mutualist relationship between the cow and the bacterium, which they do. There's a lot of bacteria that live in the cow rumen, and so they have a mutualistic relationship. But that's not, you wouldn't say they have a mutualistic with us if they're living on a cow. It's like in relationship. There's the two that are together. Does that make sense? Okay, cool. So commensal, one does nothing, the other one's happy. That's the bacterium would be happy because they're happy living in our environment, living off our diet and our cells and all of our proteins, lipids, carbohydrates. And then parasitism is the one we focus on this class. Unfortunately, it'd be cool if we could teach a whole class on the benefits of bacteria. Maybe that's down the pipe somewhere because there's so much information about probiotics now. But parasitism is what we focus on, is that we're harmed when the bacteria are living the dream. I always say live in the dream, LTD, because my girlfriend in college almost had us talked into getting LTD tattoos. So we're out drinking one night and she's like, let's go get LTD tattoos, live in the dream. I was like, yes, that is such a good idea. Thank God that did not happen. So except it'd be funnier now if I actually did it. Okay. Okay, so what else is important in a bacterial environment? So I talk about in my other class in anatomy and physiology, we talk about the 5 requirements for life. Food, nutrients, oxygen are food, water, oxygen are the 3 main ones you might think about. But we forget that humans have a very narrow range of temperature we have to live within, and we forget humans have a very narrow range of atmosphere we have to live in. Of course, we never think of atmosphere. Why do you think of that? Well, we can't go down below the ocean without proper gear, and we can't go up into outer space without proper gear. There's a pressure that our blood vessels are happy at. And so that's what we talk about in anatomy and physiology. But the same is for bacteria. Bacteria have all of these environmental requirements depending on the genus and species, right? So depending on the species of bacteria, they have different requirements because they live all over the world. So because some live on Mount Everest, they're okay with cold. Because some live underneath the ocean, they require different sugars to survive. They require different nutrients. So Here are some terms that and I'll show you the ones that you need to know. But temperature, a range of temperatures, oxygen, a range of oxygens. This is what we're gonna focus on today. pH, there's a pH, so some are happy, there are a resident population of bacteria that live in the stomach that have to deal with a high pH, or in the vagina that have to live in a high pH. There's some bacteria that have to live in the eyes with a high salt content, or on our skin with a high salt content. But there's some that will die if they're on our skin because our skin is too salty. So they are in different environments. Radiation, some can live in toxic, like nuclear radiation storage facilities. Other gases, so some can live on methane, some can live on other types of gases. Different pressures, some can live deep below the ocean or up high in altitude. So here's the ones that we're gonna know. We're gonna know thermophiles and mesophiles. So thermophiles live in hot environments, mesophiles live within normal human range of environments. So why do we care? Well, when we get a fever, if the organism is not a thermophile, it'll die. If it's not 98.6 and our temperature goes up to 101, 102, the organism will die. Also, this is how we can treat some bacteria that don't form endospores. We can cook things, we can pasteurize things, right? Remember pasteurization? We can pasteurize things and cook a lot of those organisms out. So a thermophile likes hot environment, a mesophile is normal like us, we're mesophiles, and then a psychrophile is cold, but we're not gonna know that because we don't really talk about those very often. Aerobic, facultative, anaerobic, and anaerobic, we're gonna talk about this to death, so don't worry about it right now. We're gonna talk about it today, and we'll talk about it you'll sleep and hear me talking about that in your sleep. So acidophiles, neutrophils, and alkalophiles. So we talk more about acidophiles because they live in the acidic environments. We don't really have a lot of alkaline environments in and around our bodies, so we don't talk about that as much. But neutrophils, that's a neutral pH. Halophiles, you'll hear the term halophile a lot in the lab. We're gonna grow halophiles. So we'll grow bacteria that can tolerate a high salt environment. And then there's non-halophiles that can't tolerate salt. These other ones, those are just kind of funky. We don't really talk about those. I just thought it was interesting to know. Okay. So what do they eat? So I just kind of did this very light, and I just want you to know it on a very, very easy elementary level. Just kind of run through it. Don't stress about it too much. But, so we're talking about trophic categories. So troph is, I forget what that word means, the origin of that word. Somebody knows, throw it out there. But it's eating. So, like a lithotroph, so some bacteria only can eat inorganic substances. So they eat inorganic material like methane, ones that can live off methane, or ones that can live off sulfur in the bottom of the seabeds. Organotrophs, that's what we are. That's what every organism that we're gonna deal with, and most organisms are organotrophs that live off organic materials like glucose that we'll talk about today. Autotrophs, they're self-feeding, so like photosynthesizers feed themselves. Heterotrophs, that's most of them eat food from the outer environment. We don't really talk about photosynthetic bacteria in this class, even though I wish we would because it's cool. So troph means to eat. So phototrophs use light for food. Chemotrophs use chemicals for food. Most of us are chemotrophs. We eat chemicals. You would think like not the term chemical, but we live off of chemical environment. You know, the chemical environment. It doesn't mean like toxic chemical. Okay, so it means really like organic chemicals versus inorganic chemicals. So you could just run through that. Don't stress about it too much. Just as long as you kind of know what that means, that's important. Okay, majority of bacteria eat stuff from their environment. They eat organic stuff from their environment, and that's all that really matters. Okay, so chemo organotrophs, we're gonna talk a ton about glucose. So why are we learning about bacterial metabolism? One, because it helps us with identification. You're gonna use the characteristics about different, so Staphylococcus aureus eats different things than E. coli. They eat different foods, and they produce different byproducts. So if you have a test tube full of E. coli and a test tube full of Staphylococcus aureus, now you can tell the difference because we're gonna see that in the lab that they eat different things and they produce different things. So because they're different species, they produce different stuff and they eat different stuff. So we use that in the lab. Instead, you know, you're gonna look at the microscope and you're gonna be like, what in the heck is this? I don't know. Is this a gram positive, gram negative? Is it a, how would you know if it was 2 gram negatives? How would you know which one was Bacillus cereus and which one was, not Bacillus cereus, which one was Pseudomonas aeruginosa and which one was E. coli? How would you know the difference? You're not gonna know the difference under the microscope. You have to be able to test them in other ways. So we're gonna test them based on their food substances and how they digest stuff and where they live, all that kind of thing. So treatment, we need to know because, you know, if organisms can't handle high temperature, then we use high temperature to kill them. If we want to culture them in the lab, we use low temperatures to grow them in the lab. So we need to know the characteristics about bacteria to be able to handle them and how if we want to get rid of them or if we want to keep them. So we learn about it because they are beneficial. We learn about how they benefit us, so we want to be able to keep our microbiome. So we use something called prebiotics. This whole thing now came out about prebiotics. What prebiotics are is how we feed our probiotics, how we treat, how we make an environment suitable for our probiotics. And then because we need to culture them in the lab, and we do all kinds of commercial products like enzymes, anti-tumor, we're doing all kinds of cool cancer research, help with our immune system, laundry detergent, we use enzymes from bacteria in laundry detergent, wine, all kinds of, I mean, products that you would off the wall have no idea. Probably products that we have in this class right now we use bacteria to culture those products. And then it helps us learn about the human metabolism as well, because really this, you know, we use bacteria as a model organism to help us in the lab to figure stuff out. Okay, everybody ready? Metabolism. I'm gonna let you go through that. Okay. Okay, so this is glucose. Everybody stand up really quick. Shake it off. Take a big breath. Take a big stretch. Take a lot of oxygen in. Grab a hot tea if you need it. Okay, we're gonna talk about metabolism. So there's 4 steps. We're gonna learn these 4 steps, and you're gonna learn the parts of these steps, but we're not gonna learn it in a biochemistry fashion. So we're not gonna learn all of the chemicals, we're not gonna learn all of the biochemistry, we're just gonna learn the basics of metabolism, which is also cool when you get into bio class. This is human metabolism. A lot of it is human metabolism. So you can double dip. So let's talk about glucose. So glucose is our main sugar of life. And almost every organism, nearly every organism on the planet needs glucose. So where does glucose come from? Glucose comes from plants. So we have to have plants. Bacteria have to have plants, humans have to have plants. We have to get our glucose from plants. We're not going to talk about photosynthesis in this class, but when you put carbon dioxide and water, when you put a gas together with a water, together with sun, it makes sugar. Like, I don't know if that's ever like blown your mind hole before, but that totally blows my mind. That there's all this substance that's out there that you just add a little sun and it turns into something. Now you have a substance. And this substance, this one sugar, is what every single thing uses on the planet as a meal. Now we have to have it. and we have to have oxygen. So here, we have to have oxygen, and we have to have glucose. The two byproducts that we partner up with the most perfect partner in the whole wide world that makes those two things for us. Crazy, total trip. So let's talk about glucose and what we do. So the reason that we need glucose is to make ATP. So I always trip out about this because I've spent a lot of years in a classroom, many, many years. And I've learned all about glycolysis, all about Krebs cycle, all about all the things, and biochemistry, and photosynthesis. I had to learn it all. But you know what, it might be just my stupidity. I don't know if any of you guys have ever like put that together, and maybe it's just me, but that is why we breathe, and that is why we eat, is to make ATP. That's why we eat, and that's why we breathe. So oxygen, yeah, our cells need oxygen. Yes, our cells need food. But why? We need ATP, that is why. So what is ATP? Let's talk about it. So ATP is adenosine triphosphate. So have you guys heard of adenine? You've heard of adenine, guanine, cytosine, thymine from DNA. If you haven't, don't stress about it. We're gonna go over it. But one of our nucleic acids makes up something called ATP. So this is adenosine triphosphate. So here's the adenine. It's attached to a ribose. We'll talk a ton about ribose. Don't stress about it for right now. But then it's attached to a phosphorus with some oxygens, and another phosphorus with some oxygens, and another phosphorus with some oxygens. Those are called phosphates. Phosphate, phosphate, phosphate. So adenosine triphosphate, it has 3. Sometimes it has 2, so it'd be adenosine diphosphate. Or we have 3 triphosphate. So we'll talk about what it is and what it does. What it is molecular money. We have to have this. This is the powerhouse that powers, when you say mitochondria is the powerhouse of the cell, that's because your mitochondria makes ATP. Your mitochondria takes in the glucose, your mitochondria takes in the oxygen you breathe, and makes ATP with it. It takes those 2 tools and makes ATP. Without ATP, when you hold your breath and somebody would pass out or somebody would die from not enough oxygen, it's because they ran out of ATP. If somebody starves to death and they're not getting enough fuel, when they're not getting enough glucose, their body stops making ATP, and that's when people die, is because they're not getting enough ATP. So this is why. So yeah, I mean, out of all my classes, I've learned about, you learn about ATP, but just the point, the whole point. How many don't, if somebody already has it on their slide, don't look ahead, but if you already looked ahead, just don't answer for us. Let's just throw out some guesses. How many ATP molecules do you think one cell needs? Let's say per second. How many ATP do you think one cell needs? Wild guess. Well, totally wild guess. You could be way wrong.
A second?
Per second. 100. Who else has a good guess? Who else needs candy? Throw out a guess if you need some candy.
1,000.
Yes. Okay. 100. 1,000. We need some glucose. We need our brain to start using some ATP. What else? 10,000. Awesome. Per cell per second. Anybody else have a wild guess? Yeah. 100,000. Okay, let's move on. Ready? Everybody ready? 10 million ATP per cell in our body per second. Every single cell in our body needs 10 million molecules of ATP now. Our trillions of cells in our body need that much ATP. So much ATP. Our bodies need so much ATP. And so our bodies are just ATP machines. We make so much ATP because we have to have it. It's the battery. That's what it is. It's a battery. So that phosphate that's on the ADP, there's the last phosphate on there. I'll go through what that is and what it does. But it's a battery. And if you run out of ATP, you run out of the chemical, the battery power, the chemistry that's needed to run everything else in your body. So let's talk about kind of how that works. Okay, so we're gonna go ahead. Yeah, go ahead.
Quick
question.
Yeah.
How can a bacteria do aerobic cellular respiration without an organelle of the mitochondria?
Fantastic question. I'll try it. Let me just answer your question because we might not get to that. But actually, I'm going to try really hard to get to it. We're just going to stay a little later and we'll go later to lab. But don't let me stay too long because actually we have a longer lab today. But I'll try and stay on track if I can. Okay, so yes, they don't have a mitochondria. Fantastic question. So that's a great quiz question. That's a great exam question. They don't have mitochondria. What do? Let's talk about it. Okay, so there's 3 different types of bacterial metabolism, but we're not going to focus on that for a second. We're going to just focus on aerobic respiration. We're going to focus on what we do. We need oxygen to make ATP. We need glucose to make ATP. So just let's focus on number 1 for a minute. So we take glucose, we take oxygen, we breathe out carbon dioxide, we release water as urine or we use it, and then we make ATP as energy. This is our battery. Remember our battery. So when you're making ATP, you have adenosine with a phosphate, phosphate, phosphate. Phosphates are very negative. It's like PO43 minus. So you have a lot of negativity trying to shove together. And the way I talk to my other class about it is, where's that power coming from? The power is like a really tall Jenga pile. Because Jenga piles, just because of gravity, the higher you make your Jenga pile, the more likely it's going to tip over. The more negativity that you shove together here, the easier it's going to fly off to be able to make something run, to work something. So that's where the energy comes from, is that super, super strong negativity that has to be forcefully put on there so that now you have a really tall Jenga pile that can go and push something over, and turn something, or break something, or put something together. So now you have a little battery pack. Okay, so there's 4 steps in aerobic respiration. Aerobic respiration means oxygen. So this is an aerobic respiration. Aerobic is using oxygen. And the first step is what is called glycolysis. So glycolysis. So that means glucose lysis. It lyses glucose. So in this first step, what's going to happen is we're going to break up this 6 carbon. Remember, glucose is C6H12O6. You don't have to know that, just throwing that out there. There's 6 carbons. We're going to follow the carbons. So there's 6 carbons in glucose. These are all carbons. 1, 2, 3, 4, 5, 6. 6 carbons in a glucose molecule. Now we're going to use an enzyme through many steps, or many enzymes, to break this in half. Step 1, break up your glucose in half. Now, some scientist somewhere decided that was going to be called pyruvate. Now we have 2 different 3 carbons called pyruvate. Yeah?
Back to ATP real quick, the energy that kind of comes from this battery, is it essentially you put 2 negative magnets against each other and the pressure shooting off is the energy?
Yes, very good question. Yes, yeah, we do that kind of in my other classes, do a little demo of that. I love that, yeah, awesome. So now somebody decided to call these 3 broken up glucose molecule, they decided to call that pyruvate. So now we have 2 pyruvates together. We actually end up with, when we break things apart, it releases energy. I don't know if you remember that from chemistry, but from chemistry, it's an exergonic reaction. You break it up and you get a tiny, tiny bit of ATP. I think, don't quote me, I think it's 2. We get 2 ATP molecules from this little reaction.
It's net 2. It's net 2.
Net 2, because it takes 2 as well. Okay.
Takes-
So we get a little bit of ATP out of here. Now we have 2 3 carbon molecules, because here's our 3 carbons, here's our 3 carbons, we have 2 pyruvates. Now our 2 pyruvates go into this other step. This is the weird intermediary step. The reason I call it the weird intermediary step is because no 2 scientists can decide what to call it. It's super annoying. Some people call it intermediate step, some people call it connective step, some people call it the adjacent, like so many annoying names. We're just gonna call it the weird intermediary step. In this weird intermediary step, what happens is we're gonna break off another carbon. So now we have another step, we're breaking off another carbon. Now we have two carbon molecules because we lost a carbon and we lost a carbon. So those carbons are wandering over here in the ether, and I'll tell you where they go. But here we have a 2 carbon and a 2 carbon, and those are called acetyl groups. So now some scientists somewhere named them acetyls. Now we have 2 acetyl groups. What's this? So this is, those acetyl groups are gonna attach to something called coenzyme A. It's an enzyme. We draw enzymes like little Pac-Mans. Enzymes are proteins that have jobs, that do things. And we usually draw them as little Pac-Mans, just for fun. So those little enzymes, they actually will cut stuff apart, they'll put stuff together, they have functions that they perform throughout our body. So here it's attached to a coenzyme A. So we have acetyl 1, acetyl 2, coenzyme 1, coenzyme 2. They attach here, they're just little carriers. Like Dr. Treadway reminded me, is they're just little taxi cabs. These little taxi cabs, thank you, I use that all the time. These little taxi cabs attach, and they're just gonna bring them to do the job. They don't really have a job, their job is just to carry. So now you have 2 acetyl-CoAs. Acetyl groups with a coenzyme A. So now we move into the Krebs cycle. So now this is our guy, our superhero guy who figured out what is going on with, it's also called the citric acid cycle, or CAC sometimes. Sometimes I'll hear it called like the tricarboxylic acid cycle, other things. But this is the citric acid cycle or the Krebs cycle. I always like to call it the Krebs cycle because I like to give him props. So here, don't focus on this for right now. We're just gonna focus on our two carbons. Oh, let me tell you really quickly where these carbons went. Where did these 2 carbons go that broke off? So now we have a CO2 and a CO2. That's why we breathe. That's why we exhale CO2. This is why. Because we're breaking off carbons. Those carbons that are lost from that process, this is the reason why we exhale CO2, because we have to get rid of those. Because we're moving these 2 acetyl groups into our Krebs cycle. So now we have a carbon and a carbon and a carbon and a carbon. But don't forget, these carbons have hydrogens on them. We know that carbons have a lot of hydrogens. Let me show you that slide actually, so then you can you're not looking at my little dot structure. So here's the dot structure, you can just kind of look through that. Here's the actual glucose, here's the actual pyruvates, what the actual structure is. This is actually what I learned in biochemistry, glycolysis. You have to know this. That's what you have to know if you take biochemistry, all of the steps of glycolysis. We're just gonna take the 1 glucose, break it up, make 2 pyruvates. Okay, but there's a lot of steps involved and a lot of enzymes involved. So now we're in this weird intermediary step. Now we took the 2 pyruvates, now we have 2 acetyl-CoAs. And we lost some CO2s. So that's the intermediary step. So now we're moving into the Krebs cycle. So what we're doing when we're going through the Krebs cycle is we're getting these acetyl groups. So you have 2 carbons here, and these 2 carbons are the coenzyme A is lost. That coenzyme A is moving out and going to attach to another acetyl, and that coenzyme A is just the taxi cab. This acetyl group is gonna combine with what's called oxaloacetate. Oxaloacetate has a really long, annoying name, but what oxaloacetate is just another taxi cab. Just like coenzyme A, it's just a mover. That's all it is. So oxaloacetate has 4 carbons, and those 4 carbons are gonna go through the cycle and keep going around and around and around and around and around. Oxaloacetate is the taxi cab holder for those carbons. So now, what happens is when oxaloacetate meets with the acetyl group, That's what's called citric acid. Those two things, oxaloacetate plus the acetyl group, together are called citric acid. Does anybody have a drink in here, like a bottled drink? Yeah, do you have one right on front of you? I don't think I brought anything with me today.
I have a Dr. Pepper.
Look on the ingredients. Yeah, look on the ingredients.
Citric acid or,
Yes, citric acid.
yeah.
So most things that you eat and drink have citric acid in them from bacteria. Because they put this acetyl group together with an oxaloacetate to make citric acid. Citric acid is a preservative. It makes an acidic environment so that bacteria don't grow. It makes drinks acidic so that bacteria won't grow in the acid. That's why you always have to be careful with your diet too, not to eat too much or drink too much citric acid, because it makes things acidic. Okay, so we're moving around. Now what's gonna happen is we're gonna stop following the carbons, because the carbons are gonna be lost as CO2. What we're gonna follow is we're gonna follow our hydrogens from now on. So now these hydrogens here in our acetyl group, we have 3 hydrogens. We're gonna tear those off of our acetyl groups, and we're gonna form what's called NADH and FADH2. So NADH, NAD plus and FADH, these are also just taxi cabs. These are just placeholders. What we're gonna do with the Krebs cycle is we're gonna add a hydrogen, and we're gonna add a hydrogen. That's the whole point of the Krebs cycle. That's the only point. The only point is to rip off the hydrogens and add them to here. Rip off a hydrogen, add it to here.
Do we need to know what those are, or we just?
No, I'll tell you what they are, but just, you don't need to know the actual, it's like nicotinamide, I can't even remember what it is. I have a slide on it. Page 2.
We just need to know the abbreviation.
Yes, and all they are is placeholders. We're following the hydrogens now. We're done following the carbons, we lost those as CO2. Now we need hydrogens for this next step. Yeah?
In your slide, it says FAD to FADH2. Is it
It should be H.
Gaining-
It gains 1 H.
Only
This
gains
one
one,
gains
okay.
H. Yeah, I don't know why. This is just where I got off the internet. Maybe it's, I don't know, maybe there is 2. I don't think so. Okay, so NADH, in nicotinamide, adenine dinucleotide, NAD, that's what it means. And then here, flavin, adenine dinucleotide. No one will ever ask you that in your life of a scientist. So you could just know NADH, FADH2. But this is the structure, and now we're adding the hydrogen. This is the structure, now we're adding the hydrogen. Why do we care? Why do we want hydrogens? We want hydrogens because we're moving into the next step. This is the last step, and then we're gonna go to the lab. So this last step is what's called the electron transport chain. Have you guys heard of that? Electron transport chain? Or heard of oxidative phosphorylation? I like both of those terms because they tell you exactly what they are. Oxidative? No, that doesn't look right. I think it's I. Phosphorylation. This is how I always am. I can like spell phosphorylation, but I can't spell like store. Like it's the worst. I have the worst spelling, so just hang in there with me. Okay, so now we have NADH and FADH2. That's our main moneymaker that we need now out of the Krebs cycle. The whole point of the Krebs cycle was to rip off those hydrogens. We lose some CO2 in that process too. Maybe you get a little ATP, but this is our main goal, is to get those hydrogens. So this seems nutty, right? This seems crazy. Let me just explain it in a way that hopefully that you'll understand in an easy way. Because what happens here is we need oxygen as well, and this is why we breathe. So let's talk about in a human cell for a second. Let's talk about it in a human cell. So in a human cell, this is our mitochondria. This is what's called the cristae. It's the inner membrane. And you have this little section. there's these little sections all over the mitochondria, and you have a lot of mitochondria per cell. And this is 2-dimensional, it's 3-dimensional. So there's a lot of electron transport chains all over on the inside membrane of the mitochondria. Did somebody have a question? I saw a hand up. Okay, so let's zoom in right here. So what it is a phospholipid bilayer. So you have this phospholipid bilayer on the inner membrane of the mitochondria. And then you have these proteins. And these proteins are called cytochromes. And what these cytochromes are going to do is we're going to use those hydrogens that we needed so badly. So now what are we going to do with the hydrogens? With the hydrogens, we're going to rip them off. Sorry. We have 4 of these. You get the point. And one more big, important enzyme. Everybody write it down because everybody needs to know it. Our last hugely important enzyme is right here. I always kind of draw it phallically, sorry. I don't really know how to draw it without making it look like a penis. Okay, so here, these are called cytochromes. So these cytochromes, what they do is they're gonna pull, their job is to pull off hydrogens. So now the H is gonna leave and cross this membrane to here. So now we have a hydrogen here. So it ripped off the hydrogen and pumped it across the membrane. What it's doing is it's taking the hydrogen plus an extra electron. This is called a hydride. A hydride is a hydrogen with 2 electrons. Hydrogens normally have 1, right? It's gonna rip off these electrons. So now the electrons are what's gonna power this whole machine, electron transport, and all of these hydrogens are gonna be pumped across the membrane off of the FADH because it lost its other H. They're all gonna be taken off by these cytochromes and pumped across into this membrane. So why do we care? Now we have a bunch of hydrogens pumped in this space. Hydrogens with a positive, because they lost their electrons, now they're positive. And what does energy not like? It doesn't, the negatives of the phosphates don't like to be together. The positives of the hydrogen don't like to be together. Now you have energy. You have the ability to use that positive energy to run a machine. This is a protein machine. It's called ATP synthase. ACE means it's an enzyme. Synth means it's a synthesis. ATP, it synthesizes ATP, an enzyme that synthesizes ATP. Still, 80% of you will forget the name of that enzyme. Don't forget, everyone 100% on this question, on the next quiz, and on the midterm, and on the final, are gonna remember this enzyme. Yeah? It's an enzyme that synthesizes ATP synthase. So the hydrogens are what is gonna power this turbine to actually spin. It actually legitimately spins. And it's gonna spin around, and the power, just like a water wheel, the power of the spinning is what puts a phosphate onto ADP to make ATP. So it's gonna shove that phosphate on. That's what it does, is the spinning power grabs the phosphate and shoves it on ADP. Grabs the phosphate and shoves it on ADP with the power of the spinning. The spinning is powered with hydrogens. Yeah.
So where does the phosphate come from that actually gets added on?
So this is all full of ADP and phosphates floating around.
So the machine pushes them together. And that's-
Yes, pushes them together. Awesome. So when the hydrogen comes down, the hydrogens are attracted to oxygen. Now you have water. That's why we breathe. This is why we breathe. We breathe to power ATP synthase. Because those hydrogens are being pulled down and shoved onto an oxygen. We have water, which is a waste product. Great, we can pee it out, we can use it, whatever we need water for. But that's why we breathe. And this is why we breathe out CO2. And this is why we eat glucose. So that's called aerobic respiration. That's the whole point. It's also called, so cellular respiration is also called cellular metabolism. Why would it be called both? Because you are breathing and you are eating. Both in the same process is needed to make our ATP. How many ATP do we get by this whole machine? Here's our oxygen, here's our water. Oh, here's a little visual. That's a good little visual video. Watch as many videos as you can. Here. No, we're not moving on. 36 to 38 ATP. A crap ton of ATP. For each ATP synthase. For every single molecule of glucose. This little thing right here can make 35-ish ATP just with this. Because of the machinery. Because of aerobic respiration, how aerobic respiration works.
How long is that process?
Well, if it can make 10, I don't know from one glucose, that's a really good question to Google. Like from one, how long does it take to go from one glucose molecule all the way to one ATP molecule? I thought about that, it's a very good question. Okay, so we're gonna only cover aerobic respiration today. This is aerobic respiration in us is the exact same as it is in bacteria, except for the very good point that they don't have mitochondria. So what their whole electron transport chain happens on their cytoplasmic membrane. All of this whole process happens in the cytosol, and then the electron transport chain is on their cytoplasmic membrane.
That's for G-COMs and G-COMs?
Yes, for all bacteria and archaea, ATP, the whole process of aerobic respiration happens within the cell, and then on the cytoplasmic membrane is the electron transport chain. Yeah.
So the acidity is building up between the cytoplasmic membrane and the cell wall?
Yes, man, you guys are such rock stars. I really am like super pumped right now. I'm just telling you guys. You guys have great questions, a lot of great comments. This all super helps everybody. Thank you very much. So here, when these are pumped across, this little space right here, this is the peptidoglycan. This is the cytoplasmic membrane. This space right here is called the periplasm. And it's a tight space that's used to hold all those hydrogens. Yes, fantastic question. And remember how I said that a long time ago in evolution that they think that one bacteria engulfed another bacteria, that they think that this is what became the mitochondria. That bacteria, actually mitochondria also have their own DNA. In our own cells have DNA. And they think that that's what happened, was that it was a bacteria, and that bacteria had its own little structures, and really it just became the ATP machine and didn't really need to do anything else. Super cool. Yeah.
Is the hydrogen like being reused over and over and over again?
So the hydrogen, so now it turns into water, so now our body will use water or get rid of water.
Okay.
Yeah, so hydrogen, we have to continue to add hydrogen through glucose, really, because glucose, eventually we're using the hydrogens that were once a part of glucose. Yeah.
So is there a percentage of the water in our body that we drink as opposed to what's produced from synthase?
Fantastic question too. I would love to know that. If you ever Google that, let me know. Awesome, great questions. Okay, cool. So we're gonna get started right away in lab. So make sure you check off your pre-labs. Give your notebooks right away to Dr. Rosevear. Lab notebooks and note cards can go on that back table with Dr. Rosevear.
We didn't do no cards.
Oh, we didn't. You guys gotta keep me on the straight and narrow. Okay, let's do it real quick. notebook and their note card into you downstairs, is that okay? Okay. So this is Pseudomonas aeruginosa. Say it with me. Pseudomonas aeruginosa. 2 more times. Pseudomonas aeruginosa.
Pseudomonas aeruginosa.
So Pseudomonas aeruginosa is a scary one. You'll hear about this a lot in a clinical setting. If you haven't already, you will definitely hear about it. Pseudomonas aeruginosa is getting scarier, more antibiotic resistant, all those kind of things that we worry about. And you'll hear about it a lot in a clinical setting. What shape is this? A bacilli, good. Is it gram positive or gram negative?
It's red, so negative.
Negative, because it's red, right? Does it have fimbriae?
Yeah,
Does it have flagella?
yeah.
Yes. We can't tell if it has a sex pilus. We can't tell. It definitely has lipopolysaccharides, right? Is that outer membrane? We can't see the glycocalyx or a capsule layer. That's okay. That's what it'll look like in your microscope when we look at Pseudomonas aeruginosa. Okay. So go ahead and write this one down. It's a gram negative, it's a bacilli or rod, it's motile, we know that because it has a flagella. It's aerobic, so it does what we do. It has to have oxygen, like we have to have oxygen. It doesn't undergo carbohydrate fermentation. You'll understand what that means on Tuesday. It's oxidase positive and catalase positive. You'll know what that means soon. You don't have to write this down, the green exudate or the odor fruity tortillas, but you'll hear that in a clinical setting. And when we grow Pseudomonas aeruginosa in the lab and you go like this and smell it gently, you can smell it. It smells like tortillas. And they're green. It's green on a plate when it grows. And green exudate is like pus. It makes like a green pus when your patient has it. Hopefully your patient won't. Most frequently, the skin of burn patients. We worry about that a lot with burn patients, Pseudomonas infection. And respiratory in the lungs. Hospital associated pneumonia and community associated pneumonia. We'll talk about all that stuff later. Otitis media, is it ear infection? And less frequently cardiovascular, nervous, and upper respiratory. I don't know why I said otitis media, upper respiratory. That doesn't make any sense.
Question.
Yeah.
So when we're talking about most frequent, it's the same bacteria, it's just different portals of entries that's giving it.
So why would they get a burn versus a respiratory? You mean, or?
Yeah, so like when we're going through like, and I could be completely wrong,
No,
is it going into a different portal of entry, right, that's giving them pneumonia versus an ear infection?
Yeah, great question. It's just exposure really, because so that means that the tissue tropism for Pseudomonas aeruginosa can be respiratory cells, but it can be skin cells as well.
Okay.
But yeah, depending on one, like, it's an opportunistic infection as well, like Pseudomonas aeruginosa, we wouldn't normally get it, but usually how a patient will get it is because they put a ventilator down their throat and it was on the ventilator or on the practitioner's hands. But burn patients, we wouldn't normally get it because our skin is taut and tight, right? But if we have open exposed tissue, then it can just land on there, on open exposed tissue. And the reason that we think about this is because it's aerobic, it has to have oxygen. So it wouldn't really cause a lower GI issue because it can't get oxygen down there.
But it's the same bacteria. It
Same
doesn't
bacteria.
matter where it is, it's the
Same bacteria, yeah, and some strains are different, like favor different things, but just same bacteria, just consider it all the same. Yeah, great question.
Do you want us to run the otitis media on the upper respiratory?
Yeah, I think they do consider, actually, then I was just thinking about it, I think they do consider like ear, nose, throat, upper respiratory, but, yeah. But otitis media is just the external, it would be an external, they don't generally cause an internal ear infection because they need access to oxygen, so it'd be an external ear infection or anything that's on the outside, or the lungs are full of oxygen, they need oxygen there. So